Okay, now we're let's get started. So, hello, I'm delighted to have Amin Doostmohammadi from the Niels Bohr Institute, University of Copenhagen, and today he's going to be talking to us about taming active matter from order defects to autonomous shells. And so this talk is being recorded, and as usual, if there are any questions, just in the physical room, feel free to just interrupt and ask, or if you're on the Zoom audience, you could either just unmute and ask, or you could just send a message to the Zoom, to the group or chat, or just to me, and I answered. So yeah, thank you guys for coming, and yeah, so take it away. Thank you.
Yes. Hello everyone, and thanks a lot, Farzan and David, for the kind invitation. I'll be talking, as the title suggests, I'll be talking about active matter, and just few words on active matter. To me, fascinating examples of active matter are these bacterial swarms or these cellular tissues. And the common feature in all this is that the energy injection happens locally at the level of individual particles that are driving this system. And by this local injection, this at the level of individual bacteria or the individual cell, these systems are constantly driven out of thermodynamic equilibrium, and they are able to create patterns on the scales that are much larger than the scale of the individual. What's fascinating is that if we go one scale down, if we go inside the cell and look at the material, the cytoskeletal filaments and motor proteins that form the cytoskeleton of the cell, those are also active. So those have the same features, that the energy injection is local, they're constantly driven out of thermodynamic equilibrium, and they are able to create motion on the scales larger than the individual.
So
now,
Amin, may I ask a question?
Sure.
So in the previous slide, I know that people sometimes call this swarming turbulence, but in what sense is there actually an energy cascade? It looks like energy is being injected not at the large scales. It looks like there's a very low Reynolds number in practice. So turbulence just means for our purposes here, disorderly dynamics. Or
yes. So I will get to a more quantitative comparison of what kind of shared feature or differences they have. But just to answer shortly, yes. So here the turbulence is just in terms of the visual similarities and the chaotic behavior. But I think in the second part of my talk, I will mention some words about also if there are some also technical similarities between these sort of chaotic flows and the classic turbulence.
Okay, fine. We can wait. Thank you.
Yes, thanks. Yeah. So these are the natural examples, but we can also learn from nature to build our own materials that are capable of creating these collective flows. And very successful example has been these systems made of subcellular filaments, taking microtubules from inside a cell and mixing them with kinesin motors, and then having bundles of this. They also produce these collective flows. Now, these materials are much simpler, and one can ask some questions about controlling these. For example, if I take this material and put it in a square confinement, that chaotic looking flow self-organizes into one shear flow. And one can even make it more interesting and then put it in some kind of a racetrack, and then the thing starts flowing. It's kind of a self-pumping fluid. What's amazing is that then this can reach to the speeds of about millimeter per second and can persist up to meter scale, which is comparable to the some of the current microfluidic technologies. And
so another question, please. This system can go clockwise and counterclockwise with equal probability. It just depends on the initial condition, or
it will be 50-50. It's a spontaneous symmetry breaking. So actually, I don't have the video here, but we have these experiments that you put them in sort of rings, And then you can see some of the rings are going clockwise, some of them are going counterclockwise.
Thank you.
Sure. And what's also interesting about this is that then it is kind of universal behavioral. It also happens if you confine cells or bacteria, they also break the symmetry and they are able to do this kind of self-pumping. Now, with this very short introduction of the active material systems that I will be talking about in this talk, I'll just draw a kind of a big picture of why we are studying these active materials. On one hand, to me, it is because we want to use ideas from physics to learn something about biology. For example, we now know in humans when cancer invades, in more than 90% of the time it does it collectively. So there's a question of how this coordination happens. The other interesting example is during the development. So here you can see a fly embryo at the early stages of development. Each dot is essentially a cell nucleus. And these cells, again, they coordinate their motion, they create these collective flows, and then they form these nice stripy patterns. Each of these stripes would then go on and form different parts of the fly body. So understanding how these collective flows emerge is also important in this context. But as I mentioned, mentioned a little bit about this, the other reason for studying active materials is to learn from nature and to design materials that are capable of showing these properties. For example, for using sort of for micro cargo transport by bacteria, or people have tried suspending gears inside bacterial baths and looking at some kind of a mechanical work coming out of it. So this is also the structure of my talk is designed to address both these aspects. I will first start giving examples about using physics to understand about biology. And then in the second part, I will also talk about how we can imitate nature in a sense and have some control over these active materials. So the first example that I want to talk about, this is in collaboration with Kevin Foster at Oxford Zoology and Mac Durham at Sheffield and Julia Yeomans at Oxford. And the idea is pretty simple. There's this highly infectious Pseudomonas bacteria. These bacteria, they move by using these pili. They're like legs that they use to grab on surfaces and move. And in our collaborators, they make mutants of these bacteria that have more legs. And then you can check that these mutants individually move 2 times faster. Now, the question is, if this moving individually 2 times faster give any advantage in terms of invading the space. So to test this, then we have a racetrack between a wild type, that is the slow-moving bacteria, and the delpilage, which is the mutant. So the mutant, again, individually moves faster, twice actually faster. But when we look at the invasion, you can see while the fast one initially takes over time, the slow-moving bacteria actually outcompetes the fast-moving one. And we wanted to understand why this is happening. And indeed, one can ask, okay, so this is the racetrack, they are separated. What happens if we mix them together? So we can mix them together. Again, now the fast one are the purple ones. And then if you look closely, you will see, again, over time, the fast ones, they are left behind, and the slow one take over the front.
So what color, sorry, what color are the slow ones?
Slow ones are gray.
Okay.
So let's see if we can see it here. So we have the gray ones and the purple ones, and you can see over time, the gray ones take over the front, and then the purple ones are stuck behind. And actually, when we look more closely, we see that these purple ones, they get stuck in some kind of structures that we call rosettes. So what we wanted to understand is that why these kind of structures form only for the fast-moving bacteria. Because if we understand why they form, that explains why they cannot invade as efficiently as the wild type.
So are cell divisions happening on this time scale? I mean,
cell division is also happening, and that was actually our first test, but there is no defect in cell division, meaning that the cell division in both types is the same.
Okay, and so compared to the time scale of this movie, cell division times in Pseudomonas aeruginosa might be 40 minutes. So
Yes.
or roughly. So how long is the movie?
Movie was about hours.
Hours.
But
so there's several cell divisions.
About 48 hours.
So presumably some of it, but you're saying cell division rates, as far as you can tell, are the same.
Yeah, we have checked it actually quite consistently, that the cell division of the wild type and the mutant were the same. So there was no difference in terms of rates of cell division. So it really came down to the difference in the motility as far as we could check.
Okay, but
Yeah.
presumably there's also some component of the velocity at the front that just comes from excluded volume interactions associated with the dividing bacteria. That might be the same for both, but I'm just wondering whether, I guess it's called twitching motility with these pillars, is whether there's some other stuff going on in the dynamics. Maybe you'll talk about it. I can wait. I just
Yes.
curious.
So there was different things that we first thought about. One was the division. One was actually if pili cause different friction between the bacteria, and we ruled out many of those through the experimental tests that we looked at the interaction between the cells and so on. So I'm not presenting those data here, but at the end, our conclusion was that it really comes down to the difference in motility.
Okay.
And it seems like that the motility, the higher motility, is causing the fast-moving ones to form these rosette structures. That I will.
Yeah. What is the substrate? Did you treat the substrate? Is it glass? What is it?
It's glass, and there is agar on top. Yes. So the situation is the same for both of the bacteria in terms of the substrate. So so here our challenge was to explain why these rosettes form, because to us, if we could explain why rosettes form, then that would explain why the fast-moving one gets stuck in these rosettes and cannot invade further. So we started looking closely at the organization of these bacteria, and we looked at the orientation. So here you can see immediately that there are different domains with different orientations, and more importantly, in the regions where different domains of different orientations meet, then you have singularities in the orientation. And in particular, we find two types of singularities in this system that are known as half-integer topological defects. So apologies to the experts here. So we have a plus half, that the charge of the defect comes from if I take a 2 pi rotation around the core of this singularity, then the direction of my bacteria rotates by pi, and that is a plus half defect. If I take a 2 pi counterclockwise rotation around the minus half, then the orientation changes by minus pi, so that's called minus half charge. And we see these two defects that are known to exist in the phase that's called the nematic liquid crystal. So these bacteria are showing some features of the nematic liquid crystal. And that also you can see from the movie that these defects, they get created constantly and continuously, they get created and annihilated. And normally at a steady state, you have a certain number of these defects in the system. So we started by a simplest possible model that we could drive for this system, and that was this self-propelled rods, essentially a bunch of rods that are interacting only via volume exclusion, and they have force on them. And that's governed by just.
I mean, can I ask a question?
Sure.
But I guess it's irrelevant both for the model and for the experiment. So what's the distribution of length of the bacteria, both the mutant and the wild type? And from the picture for the model, it looks like it's all monodispersed, but is that is that controlled in the experiment, or do you know what the distribution is?
I'm not sure if we checked for the distribution. As far as I remember, it was more or less monodispersed, but the mutant was a little bit longer compared to the wild type.
Okay.
Yeah, that we haven't taken into account in this model. Yeah.
Okay.
But
Presumably, that if they're dividing on this time scale, maybe they're not in this particular slide, they're getting longer before they divide. So there'll be some monodispersity just if they're around long enough to grow twice as long and then divide, right?
But we are interested in the dynamics of the front, and over the time scale that is longer than the division time scales.
The time scale is longer or shorter than the division time?
Longer than the division time scale.
Okay.
Yeah.
Okay.
So
Thank you. And this is not very well ordered. It's lots of defects, so it might be local nematic order, but there's presumably not much long range nematic order. If you measured that.
No, I don't think there is a very long range nematic order. In principle, actually, I'm not sure if you can get long range in this kind of system. It will be just quasi long range.
Okay.
Yeah.
Okay. But you certainly have a high density of defects, and they look unbound, approximately, at least to me.
Yes. So the defects are unbound, and I'll actually also come back to that.
Thank you.
Yeah. So, but the idea with this model was just the simplest possible. So it has only force on the bacteria and volume exclusion. It looks qualitatively doing the same thing, forming these plus minus half defects. They get created in pair, annihilated, and you get at steady state certain number. But more importantly, we can compare directly the velocity field around these defects between the experiments and the model. And here you can see the difference between the minus half and the plus half. The minus half is symmetric flow structure, both in the experiments and in the discrete model. But the plus half has a broken symmetry because of the shape, and the flow has also broken symmetry, meaning that the plus half has kind of a self-propulsion in these systems. It is moving towards the head. Now, this is from the discrete model. Can we say something from the continuum model? There is a very good continuum model for describing these systems with nematic topological defects, and that's essentially a model of active liquid crystals. To do that, we consider the orientational order parameter as a nematic tensor. So this has a time derivative and advection. On the right-hand side, as usual, we have the molecular field, just the relaxation towards the minimum of free energy. That includes also the resistance to deformations. So we can have two modes of bend and splay in this system. So this is taken into account into molecular field. And if you have elongated particle, in addition to moving with the flow, the particles should also respond to gradients in the flow. So if there's vorticity, they rotate. If there's shear, they align with the shear. And that's taken into account with this so-called co-rotation term. And this is coupled to the mass and momentum conservation without the active stress. These are exactly equations for passive liquid crystals. But one can show that in order to consider the active stress, the dominating contribution becomes proportional to the nematic order. This one can show mathematically by considering loss of stresslets and coarse graining over them. But physically, this just means that since this is a stress, divergence of the stress gives force. This means any gradients in Q in the nematic order create flow in the system. Now, the advantage of this continuum description is that I can prescribe the director field, the orientation of the defects, put it in this equation, and then using Green's function, I can analytically calculate the velocity field around these defects and compare it with what we had with the discrete model and with the experiments. And this is what you expect from the analytic description. But again, the important thing here is that then you can see that the plus half defect is motile. It is moving towards the head. And more importantly, from the continuum theory, we expect that the plus half defect should move faster for the fast moving bacteria. And this is checked in the experiments. Now we have the mini square displacement for the minus half defects between the wild type and the mutant. They are the same. They essentially have the same kind of dynamics, but the plus half defect has this ballistic motion and is much faster in the fast moving bacteria compared to the slow moving one. So at this point, then we had an idea that what happens in the fast moving bacteria is that these 2+½ defects, these comet-shaped defects, can come very close to each other because they're very motile. And that would essentially form a aster-like full integer defect. And we know from the theory of liquid crystal that if you have a full integer defect in a 2D layer, that is unstable. The process of escape to the third dimension should occur. So essentially, bacteria in the fast-moving strain would stand up when these two defects come very close to each other. That we can check in the simulations from the rods. We can shoot two defects towards each other and allow also the escape to the third dimension, and we can see that some kind of a reverse domino effect occurs when we shoot these towards each other. And more importantly, you can see it in the experiments that whenever these two defects come together, bacteria get out of focus, meaning that they stand up. It happens also in the mixture that the fast-moving mutants, they stand up when these 2+½ defects come together.
So I mean, that's very beautiful. Can I just ask a couple of questions about, if you could go back to hydrodynamic equations again, just a couple quick questions there. Yeah, that one. So if I look at this thing, I would have thought there would be some a drag, some friction in the final bottom equation with the substrate that wouldn't conserve momentum. It wouldn't be the divergence of something. Then shouldn't that be important? These are on a hard agar plate or something.
That's a very good point. So these equations in this form, they don't include friction. But again, that's also a question that I will come back to later on in my slides. I will also talk about friction. It will not affect the shape of the velocity field around the defects, right?
I would have thought it would have some effect. There would be some frictional length associated with that drag.
Yes,
and,
but yeah, but I think in the upcoming slides, I will then include the effect of friction, and I will comment on that.
Okay, and another question about this slide is what understanding is there of zeta? Do you even know the sign of it? Can you calculate it from some more microscopic theory?
So the sign of the zeta for the systems that are extensile, meaning that they are pushing whatever fluid is from, they're pulling it from the sides and pushing it along the direction of elongation. Okay. This zeta has a positive sign. And in those systems, we expect the defects to move towards the head.
Okay.
So if for the contractile systems, zeta is negative, and we would expect the defects to move towards their tail.
Okay, but you can see
the fact that we see in these bacteria that the defects are moving towards the head in the experiments, that kind of indicates that the defects, that these systems could be extensile. But it could be even more complicated than that. I will also comment on that a little bit later in the talk.
Okay, and then another question is can we get the order of magnitude of zeta? Is there a back of the envelope estimate of what it is? We can do it for different systems. I don't have it on top of my mind, but certainly for like microtubules or so, that people have tried. We have also tried doing that kind of estimate, but I don't have the number on back of my mind. Okay, and then finally, when these two comets collide and you get to escape in the third dimension, you could show that movie one more time. It's another one or two slides ahead. Yeah, this one. So there, it looks like they're crystallizing in the core there. Okay, is that important?
It could be important. It is not important for the purpose that we were looking at, because we were trying to explain why they are not able to invade as much. And then the explanation that we have at the moment is essentially there is some kind of speed limit. If they go faster than a certain amount, then they get trapped in these kind of structures that they create themselves. And those, and then when they stand up, those are very stable. They don't break up.
Right. In fact, presumably, if there's local crystal order, there's a shear modulus, and that might cause them to stabilize a bit. Seems to me they're crystallizing. I mean, it's a very short, it's fairly short wavelength, But it looks striking. It makes a hexagonal lattice when it escapes into the third dimension.
That's a good point. No, we didn't look at the crystallization here.
Thanks.
Yeah. Sure.
I also have a question. Is there a way to see why? So is it obvious that when these two positive defects collide that they have to jam? Because I can also imagine a scenario like a revolving door where they can just get like kind of rotate next to each other and then get past each other.
Yeah, so that's exactly what happens in the wild type or with the slower velocities, that the two defects, they cannot really come close and they have a repulsion, so they rotate around each other and they move past each other. So that's why if we do these kind of simulations with the slow moving, they actually move past each other.
So is there a threshold velocity?
There is indeed a force threshold that results in this, behavior.
In the same spirit as Grace's question, if there's an offset, an impact parameter for two colliding comets, if they're offset a little bit, then they tend to go around each other and less likely to form this escaped structure.
Yes, and also it depends on the force that is acting on individual bacteria, essentially how active they are. So that passing a certain threshold, then having two defects coming together, that will result into the escape to the third dimension. Essentially, the distance of interaction is set by how active they are. And then there is also the elastic resistance to coming closer to each other.
Clarification. So in the modeling, the difference between the slow-moving bacteria and fast-moving bacteria is you set zeta to be twice or large or something?
In the continuum, it is a zeta. In the discrete model, it is a force on individual bacteria.
So you just set the force to be twice as large?
Yes. Yeah. So that, because it's an overdamped system, so larger force means larger velocity for them individually.
Can I
Yes.
ask a question? So
Yes.
those defects that they come and they hit each other, it doesn't happen in active kinematic, right? It does happen in this bacterial system.
It also happens in the continuum active pneumatics if you allow the director to go to the third dimension.
Okay. So in the experiment, it's possible, right?
Yeah, in the experiment it's possible. In the discrete model, it's possible. In the current continuum, we just use it to look at the velocity of the defects. But there are some recent work that you can see if you allow the escape to the third dimension, then you can also see in the continuum model that the director would escape to the third dimension. But this is also known from the passive liquid crystal theory in general, that if you have a circular confinement and you have 2 plus half defects, then normally this escape to the third dimension happens. And the reason for that is that you can write down the energies for these defects, and you can see that that one full integer defect has more energy than 2 half integer defects combined for the pneumatic system.
And then in the related question, perhaps in the hydrodynamic theory, do you typically work in the one Frank constant approximation just for convenience, or can you?
Yeah, for convenience. We are actually doing some work recently on the concept of the cells, and we see that actually changing the elastic constant can also have big consequences, not on the flow field, but on the stress patterns that are created around the defects.
So if K1 were not equal to K3, and
yes.
I can imagine that anisotropy would be very large in, say, a microtubule system, you could see some evidence for that in the experiments?
Yes, I think there are some experiments from Di Pablo's group in Chicago that they have actually looked into that, and they concluded that there was some anisotropy in the elastic constant. And you can look, for example, at the shape of the defects and conclude about the anisotropy of the defects. Yeah.
Thanks.
Yes. Yes. So I need to speed up a little bit if I want to talk about everything. So the second part, I'm going to talk about a completely different system.
So just don't worry too much. From our end, we have a soft boundary at 2. You could spill over.
Okay, that's good.
Right.
Yes. So the second part, I'll talk about a different system, but this is again talking about using physics ideas in biological systems to learn something about biology. And now we are looking at the epithelial cells. These are the cells lining the surfaces and cavities in our bodies. And in these systems, we also find these half-integer defects. And more importantly, we see from the experiments that cells are killed essentially, and they are expelled at the head of these +half defects. And we observe this for different cell types, from kidney cells to aggressive and non-aggressive breast cancer cells. And this is how it looks like under microscope if you look at the system. So these white spots, so the system, the cells are moving around, they are dividing, but there are also points that they are being pushed out of a monolayer. It's a process called extrusion, which is extremely important in terms of keeping the integrity of the tissue. And it appears that if we map the direction of the cells based on the direction of their elongation, then at the head portion of these +half defects, we observe many majority of the extrusion events. And we wanted to understand why this happens.
Now,
So again,
sorry, I mean, is it an overwhelming majority or is it just a
it's
slight?
about 70%.
70, and then you see a anti-extrusion tendency at the -1/2 defect. So what happens there?
No, so in the initial work that we did not observe any depletion or any also extrusion at the -half defect. We are working on some results now that it looks like that the substrate that cells are on can affect these things. So if you go to soft substrates, then it seems like that you would also see extrusion at -half defects, which we don't have explanation for that now. Yeah.
And you always find a few extrusion events, perhaps that would be away from the defect.
Yes, there will be some also away from the defects. Yes, but
Thank you.
the overwhelming majority, about 70%, they are happening at these +half defects. And again, we can compare the flow field with the continuum model from these different experiments. They look similar and they look also extensile towards the head. But more importantly, in these systems, we can measure stress in the experiments. And what particularly we do is that we look at the isotropic part of the stress. This tells us how much compression, so the blue part and the negative isotropic stress is the compression, and the red and the positive part is the tension. And here, then you can immediately see the difference. At the head portion of the +half, where we observe the extrusion, we have this large compression region, both in the experiments and also in the continuum model. And indeed, you can see in the experiments that this compression builds up before the extrusion happens. So we reformulated this process without these cellular systems, they are also active, and we have the formation of + and -half topological defects. The cell happens to be at the cell. We initially thought the compression basically pushes the cell out. It turned out to be more complicated biologically. Essentially, the compression first kills the cell, and the cell immediately loses contact to the surrounding and almost simultaneously is pushed out of the monolayer.
There was a question.
Yeah. Can you go to the previous slide?
Yes.
No, sorry, one slide. Okay. So here, how did you measure the compression here?
So what you do in the experiments is that you use this technique called traction force microscopy. You have the beads in a substrate that the cells are seated on, and from the displacement of the beads, you can measure the forces, and then you can infer stress from there. So you have the whole stress tensor, and from the whole stress tensor, if you take the trace, then you calculate the isotropic part of the stress. Yes, and then from that isotropic part, if it is negative, that corresponds to compression. If it is positive, that corresponds to tension.
But you have to have a soft substrate for this nice measurement technique to work. And is it clear that the results on a harder substrate would be the same for extrusion, for example?
No, that's also what I mentioned. I think what we are seeing recently in the experiments is that depending on the stiffness of the substrate, you can have also extrusion happening at different points, like at minus
Okay.
half defect. Yeah.
Thank you. Thanks for reminding me.
Yeah. Now, when you think about this cellular system, so it shows some kind of similar behavior as with bacteria, but it is not really the same because the cells are constantly changing shape. In the bacterial system, we had a system that was composed of individual particles that were rod-shaped. So they had actually well-defined orientational order. For cells, they are actually mostly circular, and because of interaction with each other, they can change shape and they can get deformed. So we wanted to understand how these nematic features emerge in this kind of deformable system. And again, for answering this question, we went back to a discrete model, and we used the so-called phase field model. You can essentially think about it as active deformable droplets. So we have a phase field order parameter. This is 1 inside each cell, and it is 0 outside. Without this velocity field here, it would be just simple Canallen equation. The right-hand side is again relaxation towards a minimum of free energy. This gives the surface tension, and based on the definition of the free energy, can have cell-cell repulsion and gives the properties of the interface of each cell. But we can drive this system out of equilibrium by including this advection term. And what is nice about this is that then the velocity of each cell is determined from a force balance. So we have a friction that is balanced by a force, and the force we can write as a stress. So all we need to do is to define the stress tensor.
So sorry, another question. I mean, it looks like you're assuming incompressible velocity field. You're taking the velocity out in front of the gradient there. And that's maybe approximately true, but if there's extrusion into the third dimension, you'd think there would be sources and sinks, and so there would be a non-zero divergence.
Yes. Yes. So you're absolutely right. What we are doing here is that with this model here, this is completely 2D at the moment. So we are interested in seeing if we can form the defects in the system that is composed of essentially circular shaped particles.
Okay.
And if it shows the same kind of velocity field and stress field. At this level, with this equation, it's completely 2D, and we are not considering the extrusion process to happen explicitly. We have recently extended this into 3D. And over there, then you need to include, you're absolutely right, then you need to include the velocity inside the divergence operator.
Thanks.
Yes, but here it is completely 2D, what I'm talking about. Yeah. So all I need to define here is the stress, and the simplest thing that we could think about was to define a shape tensor for the cells. So based on the phase field parameter, you can define a shape tensor. The eigenvector corresponding to largest eigenvalue essentially corresponds to elongation of the direction of the cell. And similar to the active stress in the continuum model, then we have a stress that is proportional to the shape, meaning that if it gets, because of the fluctuations, it gets elongated a little bit, there's a bootstrap effect. It would get elongated more, and that will affect also the neighbors. This was the simplest approximation. We have more recently done a more direct comparison with the experiments that I will not focus on. But even the simplest formulation allows us to observe this sort of chaotic flows. But more importantly, when we look at the defects in the system, we can see that we observe the same stress patterns around the plus and minus half topological defects. And we see the same sort of compression happening at the head portion of the plus half defect. What's important for me to note here is that so far I have presented two different systems, namely the bacteria and the cells, And I have presented two different discrete models. One was a self-propelled rod model and one was the phase field model. But I have compared all these with experiments and one continuum model that was the active pneumatic model. And it seems to recapitulate all these different behaviors in these systems. So for the rest of the talk, I'm going to focus on the continuum model and ask questions about stabilization of these active systems. But before doing that, I want to make two notes about this system. One is about fluctuations. And because we know that fluctuations occur in this system. And indeed, we have recently tried to look at the continuum equations without any activity, only with fluctuations. And one can implement fluctuations on the velocity or on the order parameter. So we would have a so-called hydrodynamic noise that you see here, or we can have a rotational noise on the pneumatic order parameter. And it is expected that we would have defect formation if the noise is large enough. But if we, for example, look at a pair of defects that have been just nucleated, with the rotational noise, these are very short-lived. You can see also with some rescale time scale, very short-lived. But with the hydrodynamic noise, they actually move away. If I look at this defect pair, I can see that the plus half moves away from the minus half and gets annihilated with another minus half defect here. So qualitatively, they look different, and this is something we are looking at. But what's important for me to note is that without any activity, if we take any of these sources of noise and we look at average velocity field around the defects, we see the same kind of behavior as we saw in the experiments and as we saw with the activity. Meaning that the noise is kind of also creating this extensile-like behavior. And this was actually also indicated in some other works earlier. For example, Farzan had shown that if you have some fluctuating force that aligns with the pneumatic director, that can lead to extensile stress. And there's also some recent work recently. And we indeed see that these both types of noise can lead to same flow field and also same stress patterns. So this raises some questions about how much activity and how much fluctuations are playing a role here. And this is something we are interested in looking at currently.
So when you turn off the activity, the remaining noise is literally thermal noise, as the subscript
Yes.
KBT implies?
Yes, yes.
And can your experimental colleagues change the temperature?
Oh, that's what I'm asking them. I'm not sure how they can do it.
They're happy to do that with cells, but they're usually at 37 degrees centigrade if they're human cells. But you can imagine, at least changing temperature by 10 degrees or something and see what happens.
Would be, I think, one of the things that they can do in the experiments, and they have done, is perturbing cell-cell adhesions. And you can think of that as some kind of affecting the fluctuations in the system. If we knock down this E-cadherin that connects the cells to each other, then we would expect kind of more fluctuations in the system. That's something that they are looking at, but it is not very straightforward to change the temperature, I think.
You probably noticed, but if you do allow fluctuations in a passive pneumatic in 2 dimensions, the thermal fluctuations will drive K1 and K3 towards equality at long wavelengths.
Yes.
Might be useful.
Yes.
So they have to go to very long wavelengths to see it.
Yes. Yes. So I said that I need to make 2 points before moving on, and the other point is probably, if I don't mention, you're going to ask about it, is about a polarity. How about polarity? Because in all these continuum active pneumatics, so for example, with bacteria, you can clearly see them moving towards the head. Cells have also a polarity, but continuum description doesn't take a continuum description of active nematics doesn't take this polarity into account, but still is able to capture the behavior of defects. So how can we think about the polarity? And essentially, we can have different organization. If we have particles that are polar, or they are nematic, meaning that they have head-tail symmetry, there are different organizations they can have. And if we have a purely polar system, no boundary effect, we would expect full integer defects. The lowest energy excitations are full integer, they are vortices and anti-vortices. But with a nematic system, the lowest energy excitations are half integer defects. The fact that we see these half integer defects in bacteria and cells somehow indicates that the nematic symmetry dominates. But again, there's a question of how we can take this polarity into account. So what we have done recently is making the description much simpler. Instead of looking at a nematic tensor, now we think about polarity vector as an order parameter. So the equation becomes much simpler because now it's a vectorial equation. We have again a molecular field, just a derivative of free energy with respect to polarity, and we have similarly the coupling between orientation and flow. This is coupled with the momentum conservation, and one can include a polar force like this. So we have a force along the polarity direction, and of course also the friction can be included here. And the active stress again has to have the tensorial form that can be built from the polarity. So far, so fine. What we do is then, in defining the free energy, the normal free energy for polar system looks like this. So it's a gradient of polarity squared. So this is like what you have in Ising model, that you would want to penalize any alignment rather than alignment of the spins to each other. And this is included in this polarity term. So this penalizes any alignment rather than all the polar vectors aligning with each other. But we can add, based on symmetry, we can add another contribution to the free energy, to the elastic energy, that is based on the nematic symmetry. So we can form a nematic order parameter out of polarity and consider this term. So this term essentially penalizes all the alignments that are not anti-aligned, or they are not nematic. So what's the consequence of this? It's actually interesting. So if I look at the system without any, I will call it nematic elasticity, then the system would form the full integer defects as expected. If I look at the system without a polar free energy, it will form nematic defects. Now it is in a polar system, so this makes the description very, very simpler. But even more interesting is that if I have both elasticity, I can be in some regimes that we observe the coexistence of both full integer and half integer defects. And indeed, if you look at it, one can draw a phase diagram of the system based on the activity and what I call relative elasticity. If we have the polar elasticity dominating, the system shows only full integer defects. We have nematic elasticity shows half integer, but we have these very interesting regions where we have the coexistence. This also one can see from the charge. If I look at one line here, charge 1 corresponds to full polar, charge half full nematic, but we can have also this coexistence. This is also something we don't understand very well, and it's something we are working on at the moment, that how this coexistence can occur.
So, I mean, I have actually two questions, one about this and one about the fluctuations. So I'm not entirely sure I understand why Kp is a particularly polar term, but both terms, K and Kp in the energy, respect P going to minus P.
Yes, absolutely.
And so I would have imagined that a specifically polar term should violate P going to minus P symmetry.
But not in the deformation free energy. So that you can have. So this free energy will also have another contribution that would be like p squared over 2 plus p4 over 4.
Sure, yeah.
Yeah. And essentially, if you don't consider, as I mentioned, if you don't consider this term here, you only observe the full integer defect, which is a well-known thing for the polar system.
Yeah, that's okay. Yes, but
Yeah. So it is. I think so. As I said, I'm not sure we completely understand this coexistence thing here, but it is not about. It's not about the symmetry of this term that we are taking into account. It is more about the effect it has on the, I think, on the molecular field. Because when you take then the derivative with respect to the polarity, then that shows different contributions. That doesn't respect the p2 minus p symmetry.
I think both terms will still be then odd in p going to minus p, but that's what one would expect because it's the time derivative of the polarization which is related to the molecular field. Yeah, so I understand the point that you're saying, which is that adding this additional k term, and when k is larger or smaller, I forget which one. But anyway, when k is larger than kp, appropriately normalized, you allow for plus and minus half defects to be stabilized. So there's a string tension for the mismatch of the polarization across the half defect, and that somehow becomes very small. That allows the +1 to fractionalize into 2 plus half.
Yes, so I think the way I think about it is that if you don't have this term here, and you look at the energy of the defects, you will see that the energy scales with the charge to the power 2.
Yes.
Which would mean if I don't have this term here, then 2 plus half have less energy than 1 plus 1.
Sure. Yeah.
While if I don't have this term here, this is really like what you would have for coarse-grained Ising model, or so that the lowest energy excitations are always vortex and anti-vortex.
But there also the energy of a defect will scale as the square of the charge. That's true for all XY models, pneumatic.
Yes.
But anyways, I mean, I think it's interesting. I just don't quite. I'm not sure I entirely understand the mechanism.
I'm not sure if I completely understand it too, as I mentioned. So, but I think what we are trying to do to gain more insight on this is essentially trying to do some kind of renormalization group analysis and so on, as in some of the seminal papers that David also did, and see if we include both terms together, what is the under what situation we can have this coexistence, or we can have the
there's a paper that came much later, but might be useful to you, that John Selinger worked on, that involves 6-fold order and 2-fold order at the same time, and might be useful. But what typically comes out, and I think it's great to put these two things together and ask these questions, is that in the presence of the polar term, if you try to separate the plus minus a half defects that you might think would be there for the pneumatic energy alone, there's a string that ties them together. There's linear potential, which may be what Suraj was asking. So I think you'll find that there's a linear potential that it may be very small coefficient, but that Kp, because there's a branch cut, right? From the point of view of the gradient dipolar order, the XY order parameter, it's going to look like a branch cut, and then you're going to be stuck with a linear potential proportional to Kp, not a logarithmic potential binding them together.
Okay, and then maybe the question is, how do the combination of K and Kp tune that string tension to 0? And maybe that's what's happening.
Can you somehow?
Yeah. I have another question about your fluctuations. So the images that you showed of the flow field generated by either UKBT or QKBT, so that is sort of individual snapshots of flows around plus half defects, then you average over multiple defects.
That's average over multiple.
Okay, and then multiple over the noise as well, and average over the noise as well.
Yes, yes. It's essentially the same that we do in the experiments, that we take the defects, we rotate them to the same frame of reference, and then average the flow field around it.
Okay, so what's the origin of that? Because if you just think of a passive pneumatic sort of equilibrium pneumatic, say without hydrodynamics, then we know that the defect is not going to self-propel itself. Yeah. With background fluid, yes, we have backflow and things. So is this essentially fluctuation-induced backflow, which when you have good statistics, it looks like motility, because it shouldn't be actually.
So you are making a good point. It is what we find is that you need the backflow, so we can turn off the backflow in these simulations and do the same sort of experiments, and we don't see this effect. So it is, what I think is happening is that fluctuations, they create the defects, and once you have the defects, then the elastic backflow, the flow field that the elastic backflow creates looks like extensile stress.
Okay.
Yeah. So, but it is, you need the backflow, and it is also very dependent on the flow aligning parameter, meaning that if you change the sign of the flow aligning parameter, then the defect would be contractile.
I see.
Yeah.
I see.
Yeah.
I have another question. So do you ever find, instead of coexistence, do you ever find a parameter regime where, depending on the initial condition, you can either end up in all integer defects or all half integer defects?
Not depending on the initial condition, but yeah, essentially, if you look at the phase diagram here, for this relative elasticity being negative and very negative, you only have half integer defects.
But I'm asking, is there a parameter regime where you could get both, depending on? I think what I'm talking about is different than having a parameter regime where you can only access either or.
You are asking if the initial condition, depending on initial condition, for the same sort of parameters, starting from some initial condition, I end up with all full integer and all half integer. We haven't observed that.
I mean, it would be like a first order. If I understand Grace correctly, it might be like a first order transition or something. Or
yeah. I think so. I have to mention, so like these points you see are, these are sort of average over 20 different realizations.
But there's another thing you can do in your simulations. Maybe you've already done it with this admixture of terms and so forth. Is if you just take a boundary condition, tangential boundary condition for the pneumatogen around the edge of a circular disk, then you will have imposed a 2π rotation on your order parameter. And if it's pure pneumatic, then you'll get two defects inside the disk. And maybe you could see this string, if there is a string, and in a simple setup like that.
Yes, yeah, that's a very good point. I think we have seen something like that. It depends also on elastic constant, of course, because when you impose that kind of boundary condition, then we are imposing the +1 topological charge.
Right.
And in some regimes, then you can see that there are two defects. In some regimes, it's only one defect, even in the pneumatic.
Right. You might want to offline and talk to Grace or Farsan. They've been looking at similar things for passive things, but
yes.
You might want to set up a Zoom meeting with one or both of them.
Yeah, sure. Yeah. Yes. Any other questions on this? Yes. So now I move away a little bit from this, and I go to the second part. That is, so far I've just talked about how topological defects can be important in biology. Now, the question and the issue is that when you look at these systems, as also David mentioned, so they they look chaotic, and topological defects dynamics are also chaotic. And this is studied in the so-called active or mesoscale turbulence, and that on itself is an interesting topic. I will just mention a few words about this as I go on. Now, what
Okay, can I just ask about your human bronchial cells?
Yes.
Those are these are in vitro. The origin was from the human bronchial passages, but they're on a plate. They're on some sort of dish.
Yeah, they are in vitro. Yeah.
Okay.
Yeah, they are from Pascal Silberstein's group.
Right. And although we have a soft boundary, you probably should finish up in 15 minutes or so. Is that possible?
Yeah, I think 15 minutes.
Okay,
it is shorter, the second part. So I think.
Thanks for your patience.
Yeah, of course. No, I was also telling Farzan that this is really nice, because normally over a Zoom meetings, it's really hard to have discussions. It really becomes one talking and then afterwards few questions. This is very nice. I really enjoyed. So
thank you.
Yeah. So what we were interested in was that if we can have some sort of control over these chaotic flows. And our initial idea was considering friction, because exactly what you said, David, because what friction does, it introduces a screening length scale.
Right.
And essentially what this screening length scale means is that as you increase the friction, then the screening becomes smaller and smaller. And we have also characteristic length scale of these. If you look at these vortices, they have some characteristic length scale, which is so-called activity-induced length scale. So our idea was that if we increase friction enough, then the screening length should become comparable to this intrinsic length, and something interesting should happen. And indeed, something interesting happens. These chaotic flows, they self-organize into a vortex lattice.
Wow. So there's a question in the back, though.
What is this screening length scale? What's the meaning?
Sorry?
What is this screening length scale?
So this is essentially the ratio of the kinematic viscosity to the friction coefficient.
No, what is the meaning of that?
The meaning of it is that it describes over what length scale the hydrodynamic effects, they decay very fast. They are screened. It's like if you put a flow in a confinement, and this is the effect coming from the friction in the system.
So one way to think about it, and Amin knows this, I'm sure, is if you try to do Poisson flow through a two-dimensional channel, the effect of the walls will fall off quickly over this length. You don't get a parabolic profile, you get a plug flow profile. And so it is indeed precisely a screening length that tells the hydrodynamic interactions are not as long range as you think.
Yeah. So I think you can think about it as like a confinement, putting some kind of confinement to the flow, but by putting the friction. And so what happens is that when this length scale becomes comparable to the intrinsic length scale, then you have a vortex lattice emerging. And more interestingly, then the defects also organize into a structured pattern. And the system is still active, but then you have this sort of a network of defects emerging.
So this is an anti-ferromagnetic vortex lattice. In the sense you have defects of both sides.
Yes, it is an anti-ferromagnetic, exactly. Yeah.
In that sense, it's not like the Abrikosov lattice, which all has the same sign defects. It's a very different. No, it's beautiful.
Yeah.
Is it possible to do it in experiment?
Not this part. So I think the vortex lattices, they have been observed in the experiments on the cells and also on the microtubules, but on the defects, people have not looked at it. Actually, the vortex lattices in the experiments were what motivated us to do this study. But those were before the defect. So the people had not looked at the defects. So I'm not aware of if people have looked at the defect structures in this system. But as I mentioned, sorry.
I'm just saying, so just make sure I know what we're seeing. Low friction on the left, high friction on the right, and yes. Then the two pictures that are on top of that are the same vertical column. It's the same experiment, just looking at different things in flow field versus topological defects.
Precisely.
Okay.
Precisely. So it is like the vortex lattices, they are essentially interleaved with this ordered structure of defects.
Great.
Now, as I mentioned, we can also impose hydrodynamic screening by confining the system. So this is what we did next. So we can put the system in a confinement. So we have no slip boundaries on top and periodic on the sides. And we can play both with the height of the confinement or with the activity. So if the confinement, if the height is small, then the whole thing starts just breaking symmetry and moves and flows in one direction. If the size is large enough, then we have this sort of chaotic flow. But again, if this intrinsic length scale and the length scale of confinement become comparable, then we have this vortex lattice. What's nice about this vortex lattice is that it is dynamic. And if we look at now the dynamics of defect in this system, now the color map shows the vorticity. You have the -half defects that they are not motile, so they interact elastically with the boundaries. They are just pushed to the boundaries. But the +half defects, they start, so my suggestion is that try to follow one of these +half defects, and then you can see that +half defect, then you have a pair on one vortex, then they move to the next vortex, and by doing so, they span the whole system. And at this point, we had a couple of actually Dutch PhD students in the group that they mentioned, they reminded me of this.
Is one you'll definitely know. Willow, also known as drops of brandy, it's a real favourite for both Cayley and Scottish country dancers. The first couple turn by the right hand at the top, usually a girl, Cayley. The first lady turns second man, left hand, her partner right hand, working her way down.
Yeah, so, but you get the idea. So if you look again at this video, so essentially we have two defects dancing with one vortex. Then they change the partner, go to the next vortex, and by doing so, they span the whole system. So we call this a Cayley dance for that reason. And it was numerical at the beginning, but later on, Francesagues's group, they actually observed it also in the experiments on confined microtubules. Now coming back to turbulence, in addition to being some cute state, this intermediate vortex lattice is a state before going to chaotic flows. Meaning that if I now increase activity a little bit between this vortex lattice and a chaotic flow, I can have coexistence between regions that are ordered, they have vortex lattice, and regions that are disordered. I can quantify this by defining entropy. It's just a fancy name for the magnitude of the vorticity. So I average it over the height, and now I have a one-dimensional signal, which is ordered for the vortex lattice, and it is disordered for the chaotic flow. Now, this is one snapshot. I can look at it over time, so I can have chymograph. And then you can see I have a background of vortex lattice, and these perturbations, this disorder, are growing in time. We call this puff in analogy with the turbulence. An interesting thing is that at low activity or at very high confinement, these puffs, these perturbations, they die out. At very large activity, they go to the entire system, but there is a critical activity where the rate of this splitting and rate of the decay, they balance each other. And we have a certain amount of domain occupied by these puffs. We can measure this, call it the turbulent fraction, measure it as a function of activity, and then it follows a power law with a coefficient 0.276.
So have you tried fitting this to directed percolation?
Yes, so it is 0.276 is exactly directed percolation.
Ah.
And what is interesting is then there were two articles in 2015 on classic turbulence that they showed independently on both 2D and 3D that classic turbulence follows the directed percolation. Now we are talking about zero Reynolds number. Not only we have the critical exponent, but also the empty interval exponents, they correspond to the directed percolation. Meaning that at least the criticality in this system corresponds to the criticality in the classic turbulence. And I think I think this was the first more or less quantitative comparison between classic turbulence and active turbulence. And one can also extend the same idea to 3D. Now we confine it in the third dimension. The defects now become lines. If the height is small enough, it is essentially quasi 2D. So meaning that all the planes are the same and the defects remain lines. If we increase the height, then the defects start buckling and the flow becomes 3D. And we see that this exactly happens when the twist in the defect lines starts happening. So it's essentially a twist mode that gets activated in the system, and that corresponds to transition to the 3D flows. And it was also observed in the experiments later on that you can form these loops and you can have these twists in the topological defect lines. I just come to the last part of the talk, and that is my favorite part about stabilization with topological constraint. And we can take the same system of microtubules and make emulsion out of it. This is what they did in Andreas Bausch's lab. And then you form this beautiful living, I would call it organism, that doesn't have DNA and doesn't have anything. So it's like a living organism. It moves around, creates these protrusions, and so on. And you can imagine modeling this kind of system is a nightmare. You have hydrodynamics, you have deformable boundaries, and you have active pneumatics sitting on the interface.
Well,
But
sorry,
I think,
sorry, can you say again what's happening in the dancing thing on the right in the movie there?
so it is
What that
so,
again? Sorry.
so the way that you make the 2D system is that you put this on an oil-water interface. And these
Okay.
microtubules, they basically go to the oil-water interface to form this nice 2D layer. Now, if you make it, a droplet, if you make a droplet, then again, these microtubules go on the interface between oil and water.
I see.
And then you have a droplet that is active, and the active pneumatic is sitting on the shell. And it is
Okay,
able to
there's
create.
there's some sort of contact angle, I guess, associated with this droplet on the right. So it's really a 3D system.
It's a 3D system.
Okay.
It's a 3D system, and they can then play with the pressure inside the droplet, for example.
Wow.
Yeah.
Boy, that's beautiful.
And so this was a very, very hard to do modeling on this, but I think with this very smart student, we found a way of doing it. Again, we take the continuum equations. We now consider sort of Helfrich free energy for the interface. We have the energy of deformation, but the smart thing that Luke did was to design the free energy such that the pneumatic gets only stabilized at the interface between the two phases. And by doing so, then you can also look at the system like this. Now I have a shell of active system. You can even see topological defects here moving. You have a -half at the trough and the +half going at the protrusion. And you can basically start playing with it and get different forms, different shapes based on activity. But what is important is that we can look at the shell that is sitting on a substrate. And essentially, I come back to where I started. Again, we see two +half defects coming together. They form a +1. And then that forms a protrusion that then extends and changes the shape here. And one can see also the correlation between the defects and the curvature in the system. Now, this was again interesting in terms of modeling, but there's this recent beautiful experiments on this animal hydra. It's called hydra because if you cut any part of it and you wait about 2 days, it regenerates itself to the original shape. So you can see this spherical, this hollow sphere initially that if you wait about 48 hours, then forms these antlers and forms mouth and the feet. But what was amazing was that in Kinetic Currents group, they started mapping the orientation of actin filaments on this, and they can exactly see that the tentacles form at the +1 defect. Once they are formed, they leave behind -half defect at the trough, and essentially mouth and the foot also are formed at the topological defect point. Now, before I finish, I just want to say that this turns out this idea of shape-changing material and so on is not that new, and people thought about it about 30 years ago. So in what they called, I think, in the Terminator movie, they called liquid metal, and you can see lots of similarities between this material that is capable of self-deforming and what maybe we are moving to. Hopefully we are not moving towards making Terminator, but with that I would just say thank you to you, and we'll be happy to take any questions.
Great, thank you for the very nice talk. I know we've had a lot of questions, but any other questions from either physically or virtually? Not in the chat itself.
Well, that's fantastic. Thank you. Beautiful stuff.
Thanks.
Yes, you're saturated. So thank you once again.
Thanks again. I'll see you next week, hopefully.
Yes, thanks. Yeah, I'm looking forward to see you, David, next
Right.
week.
Very good.